Hello, welcome to Biology, the scientific study of life. Aneuploidy of six chromosomes. Non-disjunction of six chromosomes produces a variety of aneuploid conditions. Most of these conditions appear to upset genetics balance less than aneuploid conditions involving autosomes. This may be because the Y chromosome carries relatively few genes and because extra copies of the X chromosome become inactivated as pair bodies in somatic cells. An extra X chromosome in a male producing XXY occurs approximately once in every 2,000 live bears. People with this disorder called Klinefelter syndrome have male sex organs, but these or the tests are abnormally small, and the man is sterile, even though the extra X is inactivated. Some breast enlargement and other female body characteristics are common. The affected individual is usually of normal intelligence. Males with an extra Y chromosome XYY do not exhibit any well-defined syndrome, but they tend to be somewhat taller than average. Females with trisomy XXX, which occurs once in approximately 1,000 live births, are healthy and cannot be distinguished from XX females except by a karyotype monosomy births, and is the only known available monosomy in humans. Although these X0 individuals are phenotypically female, they are sterile because their sex organs do not mature. When provided with estrogen replacement therapy, girls with Turner syndrome to develop secondary sex characteristics must have normal intelligence. Disorders caused by structurally altered chromosomes. Many deletions in human chromosomes, even in heterozygous state, cause several problems. On such syndrome known as cri du chat, which means the cry of the cat, results from a specific deletion in chromosomes 5. A child born with this deletion is mentally retarded, has a small head with unusual facial features, and has a cry that sounds like a mewing of a distressed cat. Such individuals usually die in infancy or early childhood. Another type of chromosomal structural alteration associated with the human disorders is translocation, the attachment of a fragment from one chromosome to another. Non-homologous chromosomal translocations have been implicated in certain cancers, including chronic myelogenous leukemia, CML. Leukemia is a cancer affecting the cells that give rise to white blood cells. And in the cancer cells of CML patients, reciprocal translocation has occurred in these cells. The exchange of a large portion of chromosome 22 with a small fragment from a tip of chromosome 9 produces notice that this picture. Okay, back to leukemia. Cells that give rise to white blood cells, and the cancers affecting the cells that and in the cancer cells of SML patients, a reciprocal translocation has occurred in these cells. The exchange of a large portion of chromosome 22 with a small fragment from a tip of chromosome 9 produces a much shortened, easily recognized chromosome 22 called the Philadelphia chromosome. Some inheritance patterns are exceptions to the standard chromosome theory. In the previous section that we learned about abnormal deviation from the usual patterns of chromosomal inheritance, we conclude this chapter by describing two normal exceptions to Mendelian genetics, one involving genes located in the nucleus and the other involving genes located outside the nucleus. Genomic imprinting, genomic imprinting. Bon. Throughout our discussions of Mendelian genetics and the chromosomal basis of inheritance, we have assumed that a specific allele will have the same effect regardless of whether it was inherited from the mother or the father. This is probably a safe assumption most of the time. For example, when Mendel crossed parable flowered pea plants with white flowered pea plants, he observed the same results regardless of whether the parable flowered parent supplied the wither or the pollen. In recent years, however, geneticists have identified 2 or 3 dozen traits in mammals that depends on which parent passed along with allele for those traits. Such variation in phenotype depending on whether an allele is inherited from a male or female parent is called genomic imprinting. Note that the issue here is not sex linkage, most imprinted genes are on outcomes. Genomic imprinting occurs during the formation of gametes and results in the silencing of one allele of certain genes. Because these genomes are imprinted differently in sperm and ova, a zygote expressed only one allele of an imprinted gene, either the allele inherited from the female parent or the allele inherited from the male parent. The imprints are transmitted to all the body cells during development, so the same allele of a given gene, either the maternally inherited allele or the paternally inherited allele, is expressed in all cells of that organism in each generation. The old imprints are erased. A gamete producing cells and the chromosomes of the developing gametes are newly imprinted according to the sex of the individual. In a given species, the imprinted genes are always imprinted in the same way. For instance, a gene imprinted for maternal allele expressions is always imprinted for maternal alleles expressions generation after generation. Consider, for example, the gene for insulin-like growth factor 2, 1GF2, one of the first imprinted genes to be identified, although this growth factor is required for normal paternal parental growth. Only the paternal allele is expressed. Evidence that the GF21I GF2 gene is imprinted initially came from crosses between wild-type mice and dwarf mice homozygous for a recessive mutation in the IGF2 gene. The phenotypes of heterozygous offspring, one normal allele is one mutant differ depending on whether the mutant allele came from the mother or the father. What exactly is a genomic imprint? In many cases, it seems to consist of methyl CH3 groups that are added to cytosine nucleotides of one of the alleles. Such methylation may directly silence the allele. An effect consists with evidence that heavily methylated genes are usually inactive. However, for a few genes, methylation has been shown to activate expression of the allele. This is the case for the IGF2 gene. Methylation of a certain DNA sequence on the paternal chromosome leads to expression of the paternal IGF2 allele. Genomic imprinting is though thought to affect only a small fraction of the genes in mammalian genomes, but most of the known imprinted genes are critical for embryonic development. In experiments with mice, for example, embryos engineered to inherit both copies of certain chromosomes from the same parent inevitably die before birth. Whether that parent is male or female, normal development apparently requires that embryonic cells have exactly one active copy, not zero, not two, of certain genes. The association of aberrant imprinting with abnormal development and certain cancers is stimulating numerous studies on how different genes are imprinted. Inheritance of organelle genes. We end with an important amendment. Not all of eukaryotic cells' genes are located on nuclear chromosomes or even in the nucleus. Some genes are located in organelles in the cytoplasm. These genes are sometimes called extranuclear genes. Mitochondria, as well as chloroplasts and other plant plastids, contain small circular DNA molecules that carry genes coding for proteins and RNA. These organelles reproduce themselves and transmit their genes to daughter organelles. Because organelle genes are not distributed to offspring according to the same rules that direct the distribution of nuclear chromosomes during meiosis, they do not display Mendelian inheritance. The first hint that extranuclear genes exist came from studies by Carol Cornish on the inheritance of yellow or white patches on the leaves of an otherwise green plant in 1990. He observed that the coloration of the offspring was determined only by the maternal parent or the source of seeds that germinate to give rise to the offspring, and not by the paternal parent, the pollen source. Subsequent research showed that such coloration patterns or variegation are due to mutations in plastid genes that control pigmentation in most plants. A zygote receives all its plastids from the cytoplasm of the egg and none from pollen, which contributes little more than a haploid set of chromosomes. As the zygote develops, plastids containing a wide type of mutant pigments genes are distributed randomly to daughter cells. The pattern of leaf correlation exhibited by a plant depends on the ratio of wide type of mutant plastids in various issues. Similar maternal inheritance is also a rule of mitochondrial genes in most animals and plants, because almost all the mitochondria passed onto a zygote come from the cytoplasm of the egg. The products of most mitochondrial genes help make up the proteins complex of the electron transport chain and ATP synthesis. Defects in one or more of those proteins therefore reduces the amount of ATP the cell can make, and have been shown to cause a number of rare human disorders, because the parts of the body most susceptible to energy deprivation are nervous system and the muscle. Most mitochondria disease primarily affect these systems. For example, a person with the disease called mitochondria myopathy suffers from weakness and intolerance of exercise and muscle deterioration. In addition to the rare disease clearly caused by defect in mitochondrial DNA, mitochondrial mutations inherited From a person's mother may contribute to at least some cases of diabetes and heart disease, as well as the other disorders that commonly uh, debilitate the elderly, such as Alzheimer's disease. In the course of lifetime, new mutations gradually accumulate in our mitochondrial DNA, and some researchers think that these mutations play a role in the normal aging process.